So we finished off 9.3 talking about Krebs cycle, which is now, has released a couple of important products such as ATP, 3-NADH, and FADH2. And so now we are going to enter into talking about the oxidative phosphorylation. Things that have to do during oxidative phosphorylation is you have chemiosmosis and you have the electron transport chain, all of which will help to make ATP. And so remember, when you have glucose entering your body and your cell, they go through glycolysis, which breaks it down into pyruvate. Pyruvate goes through, enters the mitochondria to make acetyl-CoA. You have 2 acetyl-CoA because you have 2 pyruvate that gets made in glycolysis. Acetyl-CoA, each of them goes into the Krebs cycle, you have these releases of electrons which get attached to the electron carriers, FADH2 and NADH, which will then take those electrons into oxidative phosphorylation. So these electron carriers like NADH and FADH2, they are really connecting glycolysis and Krebs cycle with oxidative phosphorylation. And oxidative phosphorylation is going to use the energy from the electron transports in order to make ATP. So when we talk about the electron transport chain, right, this is still occurring inside of the mitochondria. Specifically, it's occurring in the cristae of the mitochondria. In prokaryotes, because they don't have mitochondria, it will happen in their plasma membrane. Cristae is just a part of the inner membrane of the mitochondria, which helps to increase the surface area so that you can have a ton of electron transport chains occurring simultaneously in order to speed up the process of making ATP. Now, most of the chain's components are proteins, which help to, do ATP synthesis. Proteins have a prosthetic groups that are attached to them, like coenzymes, that help with catalytic functions in order for the reactions to occur faster. The electrons that are carried from the NADH and FADH2, they will pass down the electron transport chain, and they help with ATP synthesis. When electrons are accepted into the electron transport chain, they actually get reduced, and they get oxidized as well, and they get passed down by electron carriers so that they get more electronegative as well. So electrons are transferred from NADH and FADH2 to the electron transport chain. Remember, the electron transport chain is what makes the most amount of electrons. And they are this is going to happen in something called complex 1. So NADH transports its electrons in complex 1 of the electron transport chain, and FADH2 deposits its electrons in something called complex 2 of the electron transport chain. Now, electrons get passed along a bunch of proteins. There's one called the cytochrome, which is pretty much going to pass the electron to the oxygen, which is very electronegative, and then every oxygen is going to pick up a hydrogen atom or a hydrogen ion, which is representing that electron that's being passed in order to make water. And so you will see water as a byproduct as well. So this happens with the last cytochrome. So again, electrons, as they're being passed in the electron transport chain, they're going to be passed by a bunch of proteins, which are enzymes. Enzymes are proteins. And one of them are called the cytochromes. And each cytochrome will have an iron atom. And they will pass on these electrons to the oxygen. The oxygen will then pick up the hydrogen ions, which will then combine to make water. then the electron transport chain is going to actually help with the assistance of making ATP. It won't itself make ATP, but it will assist with making ATP and ATP synthesis. And then the electron transport chain is also going to help the electrons not fall from a high energy level to a low energy level, but in small increments, so that it is more manageable as they are getting passed down in order to make the energy that they need. So we've seen this diagram 100 times. Remember, we started off with glycolysis, then we went into the mitochondria to enter pyruvate oxidation, went through citric acid cycle, and now we are at oxidative phosphorylation, which was where we said we have chemiosmosis and electron transport chain occurring to make ATP. So what is happening in the electron transport chain? You have NADH is going to be presented here at a very high energy level, higher than FADH2, okay? NADH is going to be part of something called a complex 1. As you can see, it's the first one that you see right here where NADH is being oxidized to NAD+, so it's giving up its electrons. And then FADH2 is occurring in complex 2, where it's also being oxidized to FAD because it's losing its electrons. Those electrons will go down the chain that you see here through the various different complexes and with the help of various enzymes until it gets dropped off at the end right here, attaches to an oxygen in order to make H2O. Oxygen is very electronegative, so it has an easy time of attracting those electrons to itself. And this is just furthering the understanding of you moving, you can kind of see the movement of electrons going down, combining with oxygen to make water. All right, so what is this chemiosmosis we're talking about? So the energy that is released when these electrons are moving down the electron transport chain is actually going to be used for a specific purpose. It's going to be used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. When you have these protons or hydrogen ions that are moving down their concentration gradient across this membrane from the mitochondrial matrix to the intermembrane space, they're going to pass through something called ATP synthase. ATP synthase is an enzyme that's going to make ATP from ADP and an inorganic phosphate. So ATP synthase is pretty much combining a broken, or not broken, I should say, but it's going to be making ATP by coupling an inorganic phosphate with an ADP. And it's going to get its energy, ATP synthase will get its energy from an ion gradient, which is, if you remember, it moves ions against its concentration gradient in order to make ATP. All right, so here you have a diagram of an ATP synthase. You can see the intermembrane space is the darker orange at the top. Mitochondrial matrix is at the bottom. So you are having hydrogen ions. They're going to bind to specific binding sites on the ATP synthase, which will cause it to rotate and catalyze the phosphorylation of ADP to ATP. Remember, phosphorylation just simply means you are adding a phosphate. So you can see here at the bottom, you have ADP plus inorganic phosphate to make ATP, as the top part is rotating because of the hydrogen ions that are attaching to it. This is an example of chemiosmosis, where you are literally using the energy in protons or hydrogen ions as they're moving down their concentration gradient in order for some type of work, in this case cellular work, to occur, which is making ATP. So when we talk about oxidative phosphorylation, you are, like I said, you're talking about two components. You're talking about electron transport chain, which occurs in the first part, and then you're ending with chemiosmosis in the second part. This kind of flow is an exergonic flow of electrons that are being passed from NADH and FADH2 in order that the protons or hydrogen ions can go across the membrane from the mitochondrial matrix, which you see in the picture, to the intermembrane space. When you talk about the hydrogen gradient that is being talked about here, remember I keep saying proton because it is you talk about hydrogen ions as being a proton. And so when you talk about hydrogen ion gradient, you're also referring to it as a proton motive force, which is basically when electron carriers are arranged in the inner mitochondrial matrix so that they can take the protons from the matrix and deposit into the intermembrane space, so that you can have a pro-motive force occurring. So you can kind of see that this is occurring with the, if you look at the hydrogen ions at the top, they are being released from the different complexes in the electron transport chain. All of those hydrogen ions will then come together through the ATP synthase. So they're going to go take them from the mitochondrial matrix into the intermembrane. Mitochondrial matrix is the darker orange at the top. Intermembrane space is the lighter yellow at the bottom. Those hydrogen ions will go through the ATP synthase, cause rotation. That rotation is going to trigger the phosphorylation of ADP to make ATP in order for ATP to be made. So here you kind of see a close-up of the electron transport chain, the electron transport and the pumping of protons, which is your hydrogen ions, which is creating a hydrogen or proton gradient across the membrane. So you are literally having the driving of these protons across the membrane so that they can go through the ATP synthase in chemiosmosis, and it can go back across the membrane in order for ATP to be made. Okay, so oops, let me go back for just a second. So here, like I said, let me present this again. Sorry guys, bear with me. So I just want to recap really quickly. So remember, oxidative phosphorylation is when you have electron transport chain and then chemiosmosis. Main thing occurring in electron transport chain is when you have NADH and FADH2 depositing their electrons, which occurs by them both being oxidized, which causes hydrogen ions to be released into the intermembrane space of the mitochondria, which is the dark orange. As they are released into the inner mitochondrial membrane, they then go through back across the membrane into the matrix of the mitochondria in order for the ATP synthase to be energized so that you can have ATP being made. And that's chemiosmosis. So, electron transport chain, where does it occur? In the mitochondria. What goes in? Electrons, hence in the name electron transport chain. What comes out? You have 6 things of water, 28 ATP, and you have no more electron shuttles being made because the electron shuttles are now depositing those electrons into there, and therefore you no longer need the shuttles to carry electrons anywhere else. So let's review. Glycolysis. Where does glycolysis occur? In the cytoplasm. You have glucose that goes in, a 6-carbon molecule. you have glucose that gets broken down into 2-pyruvate. Each pyruvate is a 3-carbon molecule. You have 2 ATPs that get made in this process, and you have 2 NADH, which are your electron shuttles that are also being made. Then we move into the mitochondria, where you have pyruvate oxidation. This is obviously, like I said, the locations of mitochondria. You have the 2 pyruvate that get made in glycolysis that goes into the pyruvate oxidation. You have 2 CO2, 1 from each pyruvate. Therefore, there are no longer 3 carbon molecules each. There are 2 carbon molecules each with an acetyl-CoA attached to them. So then you don't have any ATP that gets made in this process, and you have 2 NADH that also gets made because of the electrons that get released from each pyruvate that gets converted to acetyl-CoA. Then you proceed on to Krebs cycle, which is still in the mitochondria. You have 2 acetyl-CoAs that go into this reaction. You have 2 carbon dioxides that get made, 1 per turn. You have 1 GTP, which triggers the process of ADP and inorganic phosphate to make ATP. And you have 3 NADH and 1 FADH2 that gets made per turn. Then you move on to oxidative phosphorylation. The first part of oxidative phosphorylation is your electron transport chain, and it's still in the mitochondria. You are depositing your electrons from your electron shuttles, which is your FADH2 and your NADH. You get 6 water molecules that get made, 28 ATP, and no electron shuttles are made because they are the ones that deposited the electrons in the oxidative phosphorylation or electron transport chain specifically in the first place. So during cellular respiration, okay, this is what we have. You have glucose coming in, you have NADH being the carrier, you have deposited electrons in the electron transport chain that drives a proton motive force like your ATP synthase in order to make ATP. 34% of the energy from a glucose molecule is going to be transferred to your ATP, which is our main energy source, while this is and this is what's happening in cellular respiration, in order to make a total of 32 ATP. All right. All of the other energy that is being made from these cycles is actually just lost as heat, and that can't be used for anything. It just goes into the surrounding. So here we have kind of the breakdown of what we just talked about. You see glycolysis here, makes 2 pyruvate, also makes 2 NADH. That NADH travels all the way down to the oxidative phosphorylation. You have pyruvate oxidation, that makes 2 acetyl-CoA. Acetyl-CoA will then also, they also have electron shuttles. Then you have the acetyl-CoA, which goes into the citric acid cycle. Citric acid cycle makes more electron shuttles. All those electron shuttles, if you follow the white pathway, lead to oxidative phosphorylation, where you have the electron transport chain and chemiosmosis. And all together, you have 28 ATP that's made just by electron transport chain and chemiosmosis. That gets combined with the 2 ATP made in citric acid cycle with the 2 ATP made in glycolysis for about 30 or 32 ATP per glucose molecule. That's pretty crazy. So this is just kind of the breakdown of what we just said. I'm not really going to go into this. This is in your textbook, so please make sure you understand it, understand the breakdown of it, so that you can do well on your quiz. All right, there are 3 reasons why the number of ATP is not known exactly. Why don't we know exactly the number of ATP? Because we say about 30 to 32. One is that the photophosphorylation and the redox reactions that are in this cellular respiration, they're not directly coupled. So the ratio of NADH, or the electron shuttles, to the amount of ATP molecules that are being made are not actually a whole number. All right, so therefore you can't really exactly predict what the exact number of ATP that's going to be made. Now, depending on what electron carrier actually takes the electrons, also will vary depending on how much ATP is going to be made. And then the proton motive force, or the ATP synthase, which creates this proton force, also is involved in other activities. And so it could, one day make more ATP than another, and so that will vary as well. All right, so that's it for 9.4. Make sure that you are doing the, make sure you fill out your notes for this so that you can be ready for your quiz, and make sure you are keeping up with your outline as well.